These study notes are designed to align with the AICE Marine Science 9693 syllabus for chapters 1.1 through 1.3. Good luck on paper 11, multiple choice, and paper 21, data handling, free response. 1.1 Particle theory and bonding. The nature of matter. Kinetic particle theory. All matter consists of particles in constant random motion. States of matter. Solid. Particles vibrate in a fixed lattice, incompressible. Liquid. Particles move freely but remain close. Take shape of container, incompressible. Gas. Particles move rapidly with high energy, far apart and compressible. Atomic structure. Atoms consist of 3 subatomic particles: particle, charge, location, proton, positive plus, nucleus, neutron, neutral, 0, nucleus electron, negative, orbits, shells. Atomic number, number of protons, defines the element. Stability. Atoms are most stable when their outer shell is full, 2 electrons for the first shell, 8 for the next. Chemical bonding. 1. Covalent bonds. Atoms share electron pairs, e.g., H_2O, CO_2, O_2. These are very strong. 2. Ionic bonds. Forms via electrostatic attraction between oppositely charged ions, atoms that lost or gained electrons. Example: sodium chloride. Sodium loses an electron to become N to the power of plus. Chlorine gains 1 to become Cl to the power of minus. 3. Hydrogen bonds. A weak attraction between a partially positive, delta to the power of plus, hydrogen atom of one molecule and a partially negative, delta to the power of minus, oxygen or nitrogen/fluorine atom of another. Polarity. Water is a polar molecule, dipole, because oxygen pulls shared electrons more strongly than hydrogen. Emergent properties of water. Universal solvent. Polarity allows water to dissolve many ionic and polar substances. Density of ice. Unlike most substances, solid ice is less dense than liquid water because hydrogen bonds form a wide crystalline lattice at 0 to the power of circ C. High specific heat capacity. Water requires a lot of energy to change temperature, acting as a temperature buffer for marine organisms. 1.2 Solubility in water. Key definitions. Solute. The substance being dissolved, e.g., salt. Solvent. The substance doing the dissolving, e.g., water. Solution. The mixture of both, e.g., seawater. Salinity. The concentration of dissolved salts, measured in parts per thousand, ppt or per milli. Average ocean salinity is 35 ppt. Factors affecting salinity. Decreased salinity, precipitation, river runoff, melting ice, dilution. Increased salinity, evaporation, formation of sea ice, removal of fresh water. Hypersaline, water with salinity 40 ppt, e.g., Don Juan Pond or the Dead Sea. The pH scale measures hydrogen ion, HTO the power of plus, concentration. Logarithmic, a change of 1 picohenry unit represents a 10-fold change in acidity. Ocean pH, average is 8.1 to 8.2, slightly alkaline. Increasing CO_2 in the atmosphere is causing ocean acidification, lowering pH. Gas solubility. Gases like O_2 and CO_2 enter the ocean via dissolution and are influenced by 1. Temperature. Cold water holds more gas than warm water. 2. Pressure. Higher pressure, depth, increases gas solubility. 3. Salinity. Fresh water holds more gas than salt water. 4. Turbulence. Wave action increases the rate of gas exchange at the surface. The oxygen minimum layer. Surface. High O_2 due to photosynthesis and wave turbulence. Supersaturation. Oxygen minimum layer. 100 meters minus 1000 meters. O_2 drops drastically because there is no photosynthesis, but animals/bacteria are still using O_2 for respiration. Deep ocean. O_2 rises again because the water is very cold, high solubility, pressure is high, and there are fewer organisms respiring. 1.3 Density and pressure. Factors driving density. Density is mass per unit volume. D equals m for v. 1. Temperature, inverse relationship. As temp up arrow, density down arrow. Main factor. 2. Salinity, direct relationship. As salinity up arrow, density up arrow. 3. Pressure, direct relationship. As pressure up arrow, density up arrow. Ocean stratification, layers. Pycnocline, a layer where density changes rapidly with depth. Thermocline, a layer where temperature changes rapidly with depth. Halocline, a layer where salinity changes rapidly with depth. Important concepts. Tropical waters can have a unique surface layer that is very warm but also very salty due to high evaporation. Mixing. Surface layers, top 200 meters, are mixed by wind, waves, and currents. Thermal insulation. Floating ice prevents the ocean from freezing solid and insulates the water below, protecting marine life. Quick check for the exam. These study notes cover AICE Marine Science Chapter 2, Earth Processes. This chapter focuses on the dynamic forces shaping the ocean floor and coastlines, from the deep interior of the Earth to the surface movement of tides and currents. 2.1 Tectonic Processes. Earth's internal structure. The Earth is composed of layers based on density and composition. Crust, outermost solid layer. Oceanic crust, thinner, denser, basaltic rock, rich in iron/magnesium. Continental crust, thicker, less dense, granitic rock. Mantle, thickest layer, hot semi-solid rock that moves via convection currents. Core, dense center, mostly iron and nickel. Inner core is solid. Outer core is liquid. The theory of plate tectonics, continental drift, proposed by Alfred Wegener. Evidence included the jigsaw fit of continents, matching fossil distributions, e.g. Mesosaurus, and matching mountain ranges across oceans. Modern plate tectonics explains how plates move. The lithosphere is broken into tectonic plates that float on the viscous mantle. Seafloor spreading. New crust is created at mid-ocean ridges and moves outward. Paleomagnetic stripes. As magma cools at ridges, magnetite aligns with Earth's magnetic field. Symmetrical stripes of normal and reverse polarity on either side of a ridge prove the seafloor is spreading. Plate boundaries and features. Boundary type. Motion. Key geological features. Convergent. Moving together. Trenches, Eid. Volcanoes, Eid. Subduction zones, earthquakes. Divergent. Moving apart, mid-ocean ridges. Hydrothermal vents. Seafloor spreading, transform, sliding laterally. Fault lines, abyssal plains, earthquakes. Hydrothermal vents form at divergent boundaries. Cold water seeps into cracks, is superheated by magma, dissolves minerals, and is forced back up. As it hits cold water, minerals precipitate, forming a, quote, chimney. Tsunamis, high-energy, long-wavelength waves caused by sudden energy release, underwater earthquakes or volcanic eruptions that displace a large volume of water. 2.2 Weathering, erosion, and sedimentation. The three processes: 1. Weathering, breaking down rocks in situ, in place. Chemical, change in composition, e.g. dissolving ions. Physical, mechanical break, e.g. ice wedging, temperature changes. Organic, caused by living things, e.g. tree roots, lichen. 2. Erosion, movement and transport of weathered material by ice, gravity, wind, or water. 3. Sedimentation, the deposition, settling of particles in a new location. The littoral zone, intertidal zone. The area between the highest high water mark and lowest low water mark of a spring tide. Rocky shores, high energy, resistant to erosion, steep gradient. Sandy shores, formed by erosion of sandstone, gradual slope, constantly shifting substrate. Muddy shores, low energy, protected areas, very fine silt, little to no slope. Mudflats. Estuaries, semi-enclosed body where fresh and salt water meet. Brackish. High turbidity. Deltas, triangular deposits at the mouth of a river where water slows down and drops sediment. 2.3 Tides and ocean currents. Tides. Tidal range, difference between high and low water marks. Factors: gravitational pull of moon and sun, coastline shape, narrow channels increase range, and weather, low pressure causes tidal surges. Spring tides, Earth, Moon, and Sun are in a straight line, full/new moon. Highest tidal range. Neap tides, Sun and Moon are at right angles, first/third quarter moon. Lowest tidal range. Ocean currents. Surface currents, driven by wind and global patterns. Deflected by the Coriolis effect, clockwise in the northern hemisphere, counterclockwise in the southern. Deep water currents. Thermohaline circulation, driven by density, temperature, and salinity. Form the global ocean conveyor belt. Upwelling, movement of cold, nutrient-rich water from the deep ocean to the surface. Increases biological productivity. El Niño Southern Oscillation, ENSO. Normal conditions. Strong trade winds push warm water west, Asia, Australia. Upwelling occurs off South America, Peru. El Niño, trade winds weaken. Warm water moves east towards South America. Upwelling stops. Fisheries fail due to lack of nutrients. Peru gets floods, while Australia gets drought. La Niña, stronger than normal trade winds. Intense upwelling and cooler surface temperatures in the eastern Pacific. Exam tip. These notes cover AICE Marine Science Chapter 3, Interactions in Marine Ecosystems. This chapter explores how organisms live together, how energy flows through food webs, and how nutrients are cycled through the environment. 3.1 Ecological Interactions. Ecological interactions describe how organisms affect one another within a community. Symbiosis, a relationship between two different species living in close physical contact. Parasitism, the symbiont, parasite, benefits at the expense of the host. Ectoparasites, live on the outside, e.g., copepods sea lice on salmon. Endoparasites, live on the inside, e.g., tapeworms. Commensalism, one organism benefits while the host is unaffected. Phoresis, a type of commensalism for transport, e.g., remora fish attaching to manta rays. Mutualism, both organisms benefit. Example, boxer crabs hold anemones in their claws. The crab gets protection, stinging cells, and the anemone gets food debris. 3.2 Feeding relationships and energy flow. Producers, autotrophs, organisms that fix inorganic molecules into organic carbohydrates. Photosynthesis, uses sunlight energy. 6CO_2 plus 6H_2O, X right arrow light / chlorophyll. C_6, H_12, O_6, plus 6O_2. Chemosynthesis, uses chemical energy, e.g., hydrogen sulfide, near hydrothermal vents, carried out by chemoautotrophs, extremophile bacteria. Consumers, heterotrophs, organisms that must eat others to obtain energy. Trophic levels, the feeding position in a food chain. 1. Producers, first trophic level. 2. Primary consumers, herbivores. 3. Secondary consumers, carnivores/omnivores. 4. Tertiary/quaternary consumers, apex predators. Food webs show interrelated feeding relationships. Arrows represent the direction of energy and biomass transfer. Energy transfer efficiency. Only about 10% of energy is transferred from one trophic level to the next. Losses occur due to respiration, heat, undigested food, feces, excretion, urine, and parts not eaten, roots/bones. Endothermic animals, e.g., tuna, are less efficient than ectothermic animals, e.g., sunfish, because they use more energy to maintain body heat. Pyramids. Pyramid of numbers shows the count of individuals. Can be inverted if one large producer supports many small consumers. Pyramid of biomass shows the total dry mass. Can be inverted in the ocean because phytoplankton are eaten very quickly. Standing crop is small, but productivity is high. Pyramid of energy shows the rate of energy production over time. Always pyramid-shaped. 3.3 Nutrient cycles. Nutrients move between the biotic living phase and abiotic non-living phase. Biological roles of nutrients. Nutrient, biological role, nitrogen, making amino acids, proteins, and DNA. Carbon, basis of all organic molecules, carbs, lipids, proteins. Magnesium, essential for making chlorophyll. Calcium, making bones, shells, and coral skeletons. Phosphorus, making DNA, ATP, and bones. The reservoir of nutrients. The surface layer of the ocean acts as a reservoir. It is replenished by 1. Upwelling, bringing nutrient-rich deep water to the surface. 2. Runoff, water leaching nutrients from the soil. 3. Dissolving from atmosphere, CO_2, and N_2 gas dissolving. 4. Tectonic activity, minerals released from hydrothermal vents and volcanic ash. The carbon cycle, fixation, CO_2, is taken up by phytoplankton for photosynthesis. Marine snow, Dead organic matter and feces sink to the ocean floor, removing carbon from the surface. Sedimentation. Carbon is locked in limestone, Kako_3, or fossil fuels over millions of years. Marine uplift. Tectonic plates move sedimentary rock to the surface, where weathering releases the carbon back into the cycle. Exam tip. Chapter 4. Classification and Biodiversity. This chapter explores how marine scientists organize the vast diversity of life in the ocean, the unique roles specific organisms play in their ecosystems, and how we measure the health of these environments. 4.1. The classification of marine organisms. To study billions of organisms, scientists use a taxonomic hierarchy. This system groups life based on shared features and evolutionary history. Taxonomic hierarchy. The sequence from broadest to most specific. Domain, kingdom, phylum, class, order, family, genus, species. Binomial nomenclature, a two-part Latin naming system, genus, species. Rules. Genus is capitalized, species is lowercase. Always italicized in print or underlined if handwritten, e.g. Prioness glauca. Dichotomous key, an identification tool that uses a series of either-or choices to lead the user to the correct species name. 4.2 Key groups of marine organisms. Marine life is categorized into functional and biological groups. A keystone species is one that has a disproportionately large effect on its ecosystem. Without it, the ecosystem would collapse. 1. Plankton, microscopic organisms that drift with currents. Phytoplankton producers, autotrophs, like diatoms, silica shells, and dinoflagellates can cause toxic red tides. They form the base of the food web. Zooplankton, consumers like copepods, tiny crustaceans, and larvae, juvenile stages of fish/invertebrates. 2. Echinoderms, marine invertebrates, e.g., starfish, sea urchins, sea cucumbers. Features: pentaradial symmetry, 5-part body plan, and tube feet for movement and feeding. Ecological role: the crown of thorns starfish, Cots, manages coral biodiversity by eating fast-growing corals. 3. Crustaceans, hard-shelled arthropods, e.g., crabs, lobsters, krill. Features: hard exoskeleton, chitin/calcium, jointed legs, and a nauplius larval stage. Importance: krill are a vital food source for whales and seals. Scavengers recycle nutrients. 4. Bony fish, class Osteichthyes, 95% of all fish. Features: bony skeleton, operculum, gill cover, swim bladder for buoyancy, and a visible lateral line to detect vibrations. Scales: cycloid or ctenoid, thin and flexible. 5. Cartilaginous fish, class Chondrichthyes, sharks, rays, and skates. Features: skeleton made of cartilage, denticles, sandpaper-like scales, and gill slits, no operculum. Buoyancy: no swim bladder, they must swim to stay afloat. Ecological role: apex predators that maintain the balance of the food web. 6. Macroalgae and marine plants. Macroalgae, kelp/seaweed, use a holdfast to anchor, a stipe as a stalk, and blades for photosynthesis. Marine plants, seagrass, true flowering plants with roots and rhizomes. They act as major carbon sinks and nursery grounds. 4.3 Biodiversity. Biodiversity is measured at 3 levels. 1. Species diversity, the number of different species, richness, and their relative abundance. 2. Genetic diversity, the variety of alleles within a population, crucial for adaptation. 3. Ecological diversity, the variation in ecosystems, e.g., reefs versus deep sea. Why it matters. High biodiversity leads to stable ecosystems, protects coastlines from erosion, regulates climate, carbon sinks, and provides food and medicines like KLH from limpets for cancer research. 4.4 Populations and sampling techniques. Marine scientists use specific terms and tools to study populations. Niche, the specific job or role an organism plays, where, when, how it feeds. Limiting factors: abiotic, temperature, light, pH, or biotic, competition, disease, factors that restrict population growth. Sampling methods. Method. Description. Best for. Mark, release, recapture. Mark a sample, release, and then recapture later. Mobile species. Fish, sharks. Frame quadrat. A plastic/metal square used to define a sample area. Sessile species. Plants. Barnacles. Line transect. A tape measure where species touching the line are recorded. Rapidly checking distribution. Belt transect. Quadrats placed at fixed intervals along a line. Studying zonation, e.g., from high to low tide. Chapter 5. Examples of marine ecosystems. This chapter explores the diverse habitats found within the world ocean, ranging from the sunlit surface to the dark depths, and from rocky shores to tropical coral reefs. 5.1. The open ocean. The world ocean covers over 70% of Earth's surface and is essentially one interconnected body of water. It acts as a temperature buffer, absorbing excess heat to keep the global climate stable. Zones of the open ocean. The ocean is divided into vertical zones based on light penetration. Zone. Depth. Characteristics. Epipelagic. 0 to 200 meters. Sunlight reaches here. High productivity due to phytoplankton. Mesopelagic. 200 to 1,000 meters. Twilight zone. Enough light to see, but not for photosynthesis. Bathypelagic. 1,000 to 4,000 meters. Complete darkness. Organisms rely on marine snow and bioluminescence. Abyssopelagic. 4,000 to 6,000 meters. Near freezing temperatures, home to blind/colorless species. Benthic. Seafloor, the very bottom, inhabited by organisms called benthos. 5.2. The tropical coral reef. Coral reefs are among the most biodiverse ecosystems on Earth. Corals are cnidarians that exist as polyps. Key concepts. Symbiosis. Hard corals have a mutualistic relationship with zooxanthellae, dinoflagellates, which provide food via photosynthesis. Anatomy. Polyps use nematocysts, stinging harpoons, to catch prey and secrete a calcium carbonate calyx to build the reef. Atoll formation. Darwin-Daly Dana theory. A volcanic island sinks, subsides, while the surrounding fringing reef grows upward, eventually leaving a ring of coral around a central lagoon. Reef erosion and threats. Reefs erode when they lose calcium carbonate faster than they gain it. Bioerosion, predation by parrotfish or crown-of-thorns starfish. Ocean acidification, increased CO, _2 lowers pH, making it harder for corals to build skeletons. Coral bleaching. High temperatures cause corals to expel their zooxanthellae, turning them white. 5.3. The rocky shore. The main challenge here is desiccation, drying out, and wave action. Organisms organize themselves through zonation. 1. Splash zone. Only receives salt spray, home to lichens and periwinkles. 2. Upper shore. Submerged only at high tide. Organisms like chitons and channeled rack trap water to survive. 3. Middle shore. Exposed twice daily. Barnacles and mussels use glue or byssal threads to stay attached. 4. Lower shore. Mostly submerged. Highest biodiversity. Sea stars, anemones. 5.4. The sandy shore. Unlike rocky shores, the substrate here is porous and unstable. Low biodiversity, lack of attachment points, means no seaweed. In fauna, most animals are burrowers, ghost crabs, lugworms, razor clams, to avoid predators and desiccation. 5.5. The mangrove forest. Mangroves are salt-tolerant trees found in tropical and subtropical sheltered coasts. Adaptations of the red mangrove. Prop roots, arched roots that provide stability in shifting silt and trap sediment. Lenticels. Pores on the roots that allow gas exchange in anoxic, oxygen-poor mud. Salt exclusion. Roots filter out 97% of salt. Remaining salt is shed via sacrificial leaves. Viviparous reproduction. Seeds develop into propagules while still attached to the parent tree. Importance. Nursery grounds. Provide safety for juvenile fish. Carbon sink, mangroves store up to 4 times more carbon than tropical rainforests. Coastal protection. They act as a natural breakwater against storm surges. Would you like? Chapter 6. Physiology of marine organisms. This chapter covers the biological machinery of marine life, from the microscopic structure of their cells to the complex systems they use for breathing and maintaining salt balance. 6.1. Cell structure and microscopy. All marine organisms are made of cells, the smallest units of life. Scientists use 2 main types of microscopes to study them. Light microscopes use light and glass lenses, good for viewing living cells and tissues at up to times 1500 magnification. Electron microscopes use a beam of electrons and electromagnets. They offer much higher resolution, detail, and magnification, up to times 200,000. Key organelles and functions. Organelle, description/function. Nucleus. Contains genetic material, DNA, the control center. Mitochondria, the powerhouse, site of aerobic respiration to produce ATP, energy. Chloroplasts, plants only. Site of photosynthesis contains chlorophyll to trap light. Ribosomes, small structures where protein synthesis occurs. Golgi body, modifies and packages proteins into vesicles for transport. Cell membrane, a fluid mosaic of phospholipids and proteins that controls what enters/exits. 6.2 Movement of substances. Molecules move across cell membranes through 4 primary methods. 1. Diffusion, random movement of particles from high to low concentration. Passive. 2. Facilitated diffusion, movement of large or charged molecules through channel or carrier proteins. Passive. 3. Active transport, pumping substances against a concentration gradient using ATP. Requires energy. 4. Osmosis, the diffusion of water from a high water potential to a low water potential across a selectively permeable membrane. Osmosis in marine life. Hypertonic, higher salt outside, water leaves the cell shrivels. Isotonic, equal concentration, no net movement. Hypotonic, lower salt outside, water enters the cell, animal cells may burst, plant cells become turgid. 6.3 Gas exchange. Marine organisms must take in oxygen, O_2, and release carbon dioxide, CO_2. This is governed by Fick's law, which states that diffusion is fastest when there is a large surface area, a steep concentration gradient, and a short diffusion distance. Adaptations in fish gills. High surface area, gills have many filaments and lamellae. Countercurrent exchange, blood and water flow in opposite directions. This ensures that the water always has more oxygen than the blood, maintaining a constant diffusion gradient. Ventilation. Pumped ventilation, using mouth muscles to pump water, e.g. grouper. Ram ventilation, swimming with an open mouth to force water over gills, e.g. tuna. 6.4 Osmoregulation. Marine fish live in water that is much saltier than their blood, hypertonic. They face a constant risk of dehydration. How marine fish, e.g. tuna, stay hydrated. Drink large amounts of seawater, actively pump salt Na to the power of plus and Cl to the power of minus out through specialized cells in their gills. Produce very low volumes of concentrated urine to save water. Euryhaline species, e.g. salmon. These superadjusters can live in both fresh and salt water. They can reverse their ion pumps. In the ocean, they pump salt out. In rivers, they pump salt in and produce lots of dilute urine. Would you like to see the calculation steps for finding the surface? Chapter 7. Energy. This chapter explores how energy enters marine ecosystems through autotrophic nutrition, making food from inorganic sources, and how that energy is released by organisms to power life. 7.1 Photosynthesis. The vast majority of marine primary productivity comes from photosynthesis. This process uses light energy to convert carbon dioxide and water into glucose and oxygen. The balanced equation. Chloroplast structure. Photosynthesis occurs in chloroplasts. These organelles likely originated from cyanobacteria through endosymbiosis. Thylakoids. Folded membranes stacked into grana. They provide a large surface area for light-harvesting pigments. Stroma, the fluid surrounding thylakoids where glucose is synthesized. Pigments and light penetration. Marine producers use different pigments to capture light at varying depths. Chlorophyll, A and B, absorb red and blue light, mostly in shallow water. Accessory pigments, phycobilins, xanthophylls, found in red and brown algae. These absorb green and yellow light, which penetrates deeper into the ocean than red light. Turbidity, cloudiness of water, also restricts light. Scientists use a Secchi disk to measure how deep light can reach. The 2 stages of photosynthesis. 1. Light-dependent stage in grana. Asterisk light hits chlorophyll, photoactivation. Water is split, photolysis, to provide electrons, releasing oxygen as a byproduct. Energy is stored in ATP and reduced NADP. 2. Light-independent stage/Calvin cycle in stroma. The enzyme Rubisco fixes CO_2. ATP and reduced NADP are used to turn CO_2 into glucose. 7.2 Chemosynthesis. In the deep ocean where there is no light, the aphotic zone, primary producers use chemosynthesis. Source. Hydrothermal vents release chemicals like hydrogen sulfide, H_2S. Process. Bacteria, like Endoryphtia, use chemical energy to fix CO_2 into glucose. Symbiosis. The giant tubeworm Riftia hosts these bacteria in an organ called a trophosome. The worm provides chemicals, the bacteria provide food. Feature: photosynthesis, chemosynthesis, energy source, sunlight chemical bonds, H_2S, methane, byproduct, oxygen, sulfur and water. Location: euphotic zone, surface. Deep sea, hydrothermal vents. 7.3 Respiration. Respiration is the process of releasing energy from glucose to produce ATP, the energy currency of the cell. Aerobic respiration with oxygen. The complete breakdown of glucose, occurring mostly in the mitochondria. Anaerobic respiration without oxygen. An incomplete, less efficient breakdown of glucose that occurs in the cytoplasm. In animals, produces lactic acid, lactate. This can cause muscle fatigue and must be broken down later. In plants/fungi, produces ethanol and CO_2. Factors affecting respiration. Temperature. As temperature rises, respiration increases due to higher kinetic energy and enzyme activity. However, if it gets too hot, enzymes denature and the rate crashes. Mitochondria count. Active cells, like the red muscle of a tuna, have high numbers of mitochondria to keep up with the demand for ATP. This summary covers the key concepts of Chapter 8, Fisheries for the Future, focusing on marine life cycles, sustainable fishing practices, and the mechanics of aquaculture. 8.1 Marine life cycles. Life cycles describe the stages an organism passes through from birth to maturity. Simple versus complex life cycles. Simple life cycle. No major larval stages or metamorphosis, e.g., bottlenose dolphin. Calves resemble small adults and share a similar diet. Complex life cycle. Includes different larval forms and habitats, e.g., shrimp. Stages of shrimp: egg, right-pointing arrow, nauplius, right-pointing arrow, protozoa, right-pointing arrow, mysis, right-pointing arrow, post-larva, right-pointing arrow, juvenile, right-pointing arrow, adult. Advantages of larval stages: 1. Reduced competition. Larvae often eat different food, e.g., phytoplankton, than adults, occupying different niches. 2. Distribution allows sessile, immobile species like the giant clam to colonize new areas via free-swimming larvae. 3. Extinction resistance offspring are spread across different locations. Strategies for survival: R versus K. Feature: R strategy, K strategy. Offspring number: huge, millions. Few, parental care. Little to none. High investment. Survival rate: very low, often less than 1%. High. Examples: tuna, giant clam, whales, sharks. 8.2 Sustainable fisheries. Sustainable fishing means harvesting at a rate, the maximum sustainable yield, MSY, that allows the population to replenish itself. Key factors in population dynamics: the health of a fish stock is determined by recruitment, number of fish surviving to enter the fishery; fecundity, reproductive rate, egg production; mortality, sum of natural mortality, predation, disease, and fishing mortality, harvest, bycatch. Modern fishing technology and impacts: sonar uses sound waves to locate shoals, increases efficiency but risks overfishing. Purse seine, a net that purses shut to trap pelagic fish. High risk of bycatch, dolphins, turtles. Benthic trawling, dragging heavy nets along the seabed. Extremely damaging to coral reefs and benthic ecosystems. Factory ships, large vessels that process and freeze fish at sea, allowing fleets to exploit remote waters for weeks. Management tools. To prevent stock collapse, like the North Sea cod in the 1980s, regulators use: 1. Quotas, limits on the weight of fish landed. 2. Season/area closures, protecting fish during spawning or in nursery grounds, e.g., mangroves. 3. Gear restrictions, minimum mesh sizes to allow juveniles to escape. 4. Monitoring, satellite tracking, AIS/VMS, air patrols and logbooks. 8.3 Marine aquaculture. Aquaculture is the rearing of aquatic organisms for human use. It is categorized by the intensity of the effort. Systems of production: extensive, low stocking density, natural food, low capital, e.g., mussels on ropes. Intensive, high density, artificial feeding, oxygenation, and high technology, e.g., indoor tank systems. Case studies in aquaculture: salmon, an intensive process involving stripping eggs, freshwater hatcheries, fry/par smolt, and open sea cages for growing out. Mussels, sessile organisms grown on ropes or rafts. They are filter feeders and do not require supplemental feeding. Shrimp, grown in raceway or grow-out ponds, often associated with mangrove destruction. Environmental and social impacts. Pollution, excess feed and feces can cause eutrophication. Disease, high densities promote spread, e.g., sea lice in salmon, white spot in shrimp. Invasive species, escapees, may outcompete native stocks. Socioeconomics, can provide jobs and protein, but may also displace traditional fishers or cause land price inflation. Solutions for sustainability. IMTA, integrated multi-trophic aquaculture, growing different species, fish, shellfish, seaweed, together so waste from one becomes food for another. Rehabilitation, replanting mangroves and building artificial reefs to restore wild stocks. Sustainable feed, using plant-based proteins or fish trimmings instead of wild-caught feeder fish. This breakdown follows your rubric and the specific information from the text provided, organized strictly into the 4 sections, chapters, you need for your study. Chapter 9.1, The Oil Industry and Agriculture. The oil industry. Seismic exploration. Survey ships map the seabed using high-decibel explosive impulses. This kills fish eggs/larvae and damages the vibration detection mechanisms in fish, making them unable to find mates or escape predators. Drilling releases toxins like benzene, zinc, and arsenic into the water. These enter the food chain via bioaccumulation, the buildup of substances within a single organism. Physical damage from spills, marine mammals. Oil covers blowholes, suffocation, and destroys the insulating properties of fur in otters/seals, leading to death by hypothermia. Birds. Oil makes feathers lose buoyancy and insulation. Birds die from drowning or hypothermia. Coastlines. Oil suffocates mangrove roots, lenticels, leading to coastal erosion because the roots no longer anchor the soil. Cleanup issues. Burning oil causes air pollution, acid rain. Dispersants like Corexit are toxic to plankton and cause oil to sink to the cold seabed, where it persists for decades. Dead zones. Bacteria that decompose oil respire at high rates, removing all oxygen from the water. Agriculture and eutrophication. Leaching. Inorganic fertilizers, nitrates/phosphates, dissolve in rainwater and run off into rivers and seas. The process. 1. High mineral ions cause algal blooms. 2. Overgrowth blocks light. Plants below die. 3. Decomposer bacteria break down the dead matter. 4. Bacterial respiration depletes oxygen, creating a dead zone, anoxia, where aerobic organisms suffocate. Chapter 9.2, Renewable Energy, Sewage and Refuse. Renewable energy installations. Wind turbines. Drilling for concrete bases damages benthic habitats. Noise vibrations may disorient whales/mammals. Tidal barrages. These concrete structures alter salinity and increase turbidity, cloudiness, which can block light for producers. Physical risk. Moving turbines and wave platforms pose a collision risk to fish and marine mammals. Sewage and refuse disposal. Sewage carries pathogens like Campylobacter, which kills seals, and surfactants from detergents. Surfactants break down the protective mucus layer on fish, making them vulnerable to parasites. Plastics. Over 80% of marine refuse is plastic. It is not biodegradable, but undergoes photodegradation, breaking into microplastics via UV light. Microplastics. Particles less than 5 meters m. They act as sponges for toxins like PCBs and BPA. Biomagnification. The increasing concentration of toxins, like mercury or TBT, at successively higher levels of the food chain, severely harming top predators. Entanglement. Ghost nets, abandoned fishing gear, trap and drown 500,000 mammals annually. Chapter 9.3, Global Warming and Ocean Acidification. Global warming, enhanced greenhouse effect. Cause. Burning fossil fuels increases CO_2 trapping infrared radiation in the atmosphere. Impacts. Melting ice causes sea level rise 17 centimeters in the last century, and habitat loss for polar species. Temperature causes coral bleaching, loss of symbiotic zooxanthellae. If temperatures stay high, the coral dies, destroying the entire ecosystem's foundation. Salinity. Melting ice releases fresh water, which disrupts ocean currents and harms osmoconformers. Ocean acidification. The chemistry CO_2 dissolves in water to form carbonic acid. The result, increased H2O to the power of plus ions react with carbonate ions, CO_3 to the power of 2-. This leaves fewer carbonate ions available for corals and mollusks to build their calcium carbonate, CaCO_3, shells and skeletons. Outcome: existing shells and reefs begin to dissolve, weakening the structure of the ecosystem. Chapter 9.4 Conservation and Sustainability. Key conservation tools. IUCN Red List, a barometer of life that categorizes species, e.g., endangered, critically endangered, based on population size and range. CITES, an international agreement that bans or regulates the trade of endangered species. Appendices 1, 2, and 3. MPAs and no-take zones, areas where fishing is prohibited. These create a spillover effect where healthy populations migrate out into fished areas. Sustainability and ecotourism. Ecotourism, responsible travel that minimizes impact and provides financial benefits to local people. This gives locals a stake in protecting the environment. Stakeholders. Sustainable development, meeting current needs without compromising future generations. This includes using renewable energy, solar/wind, and locally sourced materials like adobe/clay. Invasive species control. Methods include physical removal, lionfish culling, ballast water treatment for ships, and biological controls like the carp herpes virus. This summary covers the practical skills chapter, focusing on the scientific method, experimental design, and data analysis. This is a foundational chapter for succeeding in papers 2 and 4 of your marine science course. 1. Experimental planning. Planning is the first stage of the scientific method. It involves turning an initial observation, often qualitative, into a testable hypothesis. Key concepts in planning. Hypothesis, a testable explanation for an observation. Prediction, a specific statement of what will happen in the experiment, often sketched as a predicted graph. Variables. Independent variable, the factor you change, e.g., light intensity. Dependent variable, the factor you measure, e.g., rate of photosynthesis. Control variables, factors kept constant to ensure a valid test, e.g., temperature, pH, salinity. Control experiment, an identical setup where the independent variable is not changed, used for comparison. Confounding variables. In the field, these are variables you cannot control, like weather, but must measure and record to help explain your results. 2. Choosing techniques and equipment. To get accurate results, you must use the right tools and maintain a safe, ethical environment. Controlling key variables. Variable, control method, temperature, thermostatically controlled water bath, pH, use of a buffer solution, gases CO_2 or O_2, closing parenthesis, kept in excess, e.g., bubbling air or adding hydrogen carbonate. Light, fixed distance from a light source in a dark room. Accuracy versus precision. Accuracy, how close a measurement is to the true value. Precision, how refined the measurement is, e.g., more decimal places, or how close repeated measurements are to each other. Safety and ethics, identify hazards, the object, and risks, the harm it causes. Always minimize harm to living organisms. 3. Presenting data and observations. Results should be organized so that patterns are easy to see. Tables. The independent variable usually goes in the first column. Headings must include units. Data should be recorded to a consistent degree of precision, same number of decimal places. Graphs. Line graph, used for two continuous variables, e.g., time versus temperature. Bar chart, used when one variable is discontinuous/categorical, e.g., species type. Histogram, used to show the frequency of a continuous variable, e.g., number of fish in specific size ranges. 4. Evaluation of procedures. After the experiment, you must assess the quality of your work. Sources of error. Systematic error, a consistent error that affects all results the same way, e.g., a poorly calibrated scale. Random error, unpredictable fluctuations, e.g., slight temperature changes or human reaction time. Anomalous results, data points that do not fit the general trend. These should be identified and explained, not ignored. Improving the experiment. Increase reliability by taking more repeats. Increase validity by controlling more variables or using more precise equipment. Add more values for the independent variable to see a clearer pattern. 5. Analysis and conclusion. The final step is to explain what the data means. Mathematical analysis. Significant figures. Give your final answer to the same number of significant figures as the least precise measurement used. Statistical tests. Mean, the average. Standard deviation/error. Measures the spread or variability of data. Spearman's rank/chi-squared, used to see if results are statistically significant, usually a probability of less than 5% that the result is due to chance. Drawing the conclusion. 1. Describe. State the trend, e.g., as temperature increased, the rate also increased until. 2. Quote. Use specific data points from your results. 3. Explain. Use the phrase, "this is because," to link the result to scientific theory. 4. Refute/support. State whether the results support or disprove your original hypothesis.